Science is a way of life. Science is the process that guides us to discern facts from fantasy. For those who are curious and imaginative, science can be a transformation. Ladies and gentlemen, we are here to witness science, embrace science, and celebrate science. Welcome to the Annual Research Congress of the Postgraduate Institute of Science, ResCon 2020, presented proudly for the 7th consecutive year. To start the event formally, let us all rise to the national anthem. Thank you, ladies and gentlemen. We will have to solemnly remind you that we are living through tough times. We will adhere to the strictest health guidelines to ensure the safety of all the dignitaries and participants. Thus, it would be great if we could all wear masks and practice social distancing while the event is in place. Light signifies knowledge, truth, and hope. The oil lamp represents tradition, and the event sets the ambience to the whole ceremony. Let us all rise to the occasion of lighting the traditional oil lamp. To light the traditional oil lamp, I warmly call upon the chief guest, Professor Sampath Amaratunga, the chairman of the University Grants Commission. The guest of honor of the event, Professor Upul B. Disanayaka, the Vice Chancellor of the University of Peradeniya. The honored keynote speaker of the event, Professor S.H.P. Parakrama Karunaratne, the Deputy Vice Chancellor of the University of Peradeniya. Thank you, sir. The Director of the Postgraduate Institute of Science, Professor H.M. T.G.A. Pitawala. The Dean of the Faculty of Science, University of Peradeniya, Professor Saluka Kodituappu. Thank you, sir. And finally, the Chairman of the Organizing Committee of RESCON 2020, Professor G. W.A. Rohan Fernando. Thank you, sir. Thank you, ladies and gentlemen. The Postgraduate Institute of Science, PGIS, is a national institute and one of the pioneers of postgraduate education and research in Sri Lanka. To tell the world what we do as an institute and what RESCON 2020 is about, the team RESCON produced a synopsis. I hope you enjoy this video.
The dawn of modern civilization is driven by science. Science begins with our unquenching curiosity to understand the grandeur of nature and the universe. Science is our collective learning, the uninterrupted flow of knowledge from one generation to another. The Postgraduate Institute of Science, PGIS, was established in 1996 to ensure the transfer of modern scientific knowledge to the next generation. The PGIS offers master's degrees covering a majority of fields of the sciences, and it mentors MPhil and PhD students to provide their expertise to the national development, serving at research institutes around the country. The PGIS has trained over 20,000 professionals through in-service training programs, short courses, workshops, and outreach activities. By the end of 2019, the PGIS has awarded nearly 2,900 PhDs MPhils, and MSc degrees, and 1,245 postgraduate diplomas. At present, there are 4,426 students enrolled in 28 MSc programs and 671 students in MPhil and PhD research-based programs. The academic programs of the PGIS are conducted through 11 boards of study in close collaboration with the Faculty of Science, University of Peradeniya. The Board of Study in Biochemistry and Molecular Biology aims to provide a skillful and knowledgeable graduate cohort for research and industries related to molecular biology. The newest Board of Study, the Board of Study in Biomedical Sciences, aims to produce multidisciplinary researchers in connecting medicine and science. The research of the Board of Study in Chemical Sciences focuses on applying chemical and physical properties of atoms and molecules to sustainable economic growth and industrialization. The Board of Study in Earth Sciences focuses on advancing the frontiers of fundamental and applied research and disseminating knowledge on Earth Sciences realm, aiming socio-economic inspiration for the society. The Board of Study in Environmental Science encompasses study in all scientific fields related to the environment, leading to understanding and resolving current environmental issues not only in Sri Lanka but also in the world. Research in the Board of Study of Mathematics focuses on applying sophisticated mathematical techniques into industry and applied sciences. The Board of Study in Physics focuses on its research on two key areas, namely materials and physics applied to medicine. The core research areas in materials focus on the study of solar cells, fuel cells, and batteries. Diagnostic radiology, therapeutic radiology, and dosimetry are the key research aspects in physics applied to medicine. The Board of Study of Plant Science concentrates on multidisciplinary research on current topics, most recently on greening Sri Lanka, addressing the problem of urbanization and finding solutions through a green eye. The Board of Study in Science Education focuses its research on identifying issues in the teaching and learning process to enhance students' learning through effective pedagogical approaches, thus enhancing the standard of science education in Sri Lanka. Research work related to the Board of Study in Statistics and Computer Science focuses on the broader areas of computer science, applied statistics, data science, artificial intelligence, and information technology, with a special focus on emerging research areas and innovative projects in the respective domains. The research students attached to the Board of Zoological Sciences dedicate their efforts to increase the scientific knowledge of our understanding about the animal kingdom. To strengthen harmony and collaboration among the young researchers at PGIS, the Young Researchers Forum was established. It encourages students to engage in activities related to both education and social interactions. They publish the Cicitator, an in-house journal highlighting their ongoing research and scientific reviews. PGIS organizes the annual Research Congress, RESCON, to provide a platform for students and scientists from institutions around the country to share their brand new research findings. Culminating at a total of 230 presentations under 5 themes, the RESCON 2020 is the largest of its kind in Sri Lanka. For 2 days, we will be experiencing glimpses of scientific achievement of our country's scientists. We proudly invite you for the 7th annual RESCON 2020.
Thank you, ladies and gentlemen. I hope that synopsis gave a good representation of who we are and what we do. To proceed with the next portion of our event, I cordially invite the following esteemed guests to the head table. The chief guest, Professor Sampath Amaratunga, the chairman of the University Grants Commission. So please come up to the head table. The guest of honor for this event, Professor Upulbi Disanayake, the Vice Chancellor of the University of Peradeniya, the Director of Postgraduate Institute of Science, Professor H.M. PGA Pitawala, and the Chairman of the Organizing Committee of RESCON 2020, Professor G.W.A. Rohan Fernando. Thank you, sir. Now, to welcome the gathering, I cordially invite Professor H.M. T.G.A. Pitawala, Director of the Postgraduate Institute of Science, to deliver the welcome address.
Good afternoon, ladies and gentlemen. I warmly welcome you all for the PGIS Research Congress, RESCON 2020. Professor Sampath Amaratunga, Chairman of the University Grant Commission, Professor Upulbi Disanayake, Vice Chancellor of the University of Peradeniya, Professor Parakrama Karunaratne, today keynote speaker, Professor Rohan Fernando, Chairman of the PGIS RESCON 2020, Professor Saluka Kodituaku, Dean Faculty of Science, distinguished guests, ladies and gentlemen, my dear students. It is great pleasure for me to welcome you all to the opening ceremony of Research Congress of PGIS. Actually, this is a very special day for us, very special annual event for us, and personally, it is one of the memorable day for me. Actually, we are compelled to conduct this function with a small number of gathering due to the coronavirus pandemic. However, thanks to the online platform, thanks to the technology, I believe that the opening ceremony may have a larger audience than ever before in the history of our institute. Ladies and gentlemen, this young and unique postgraduate institute has made a commendable progress during last 2 decades. It has successfully expanded over the years to conduct large number of highly demanding postgraduate programs and related activities. At present, more than 2,300 postgraduate students from different universities in Sri Lanka and from abroad are following our programs. Also, the teaching staff of our institute is drawn from several universities and institutes of our country. It is indeed such an excellent staff representative of whole country. Chairman, sir, I would like to draw your attention regarding what I mentioned on our staff members. This research congress is organized, directed by Professor Rohan Fernando. He is attached to the Open University of Sri Lanka. One more example, several staff members of your university are in the teaching panel of our institute, including the Dean, Faculty of Graduate Studies. So I hope that you can understand the strength of our teaching staff. During this year, our institute has worked hard, even in the middle of coronavirus, to promote new research opening and the quality of teaching. I would like to thank all the staff on that regards. Our hard work has achieved significant results that we are happy and really proud of our institute. This year, we produced more than 250 MSc graduates, around 40 PhD and MPhil graduates, and hundreds of postgraduate diploma graduates. Ladies and gentlemen, I take this opportunity to extend my deep appreciation to the University Grant Commission of Sri Lanka for supporting the effort for improving our institute, PGIS. I want to especially thank Professor Sampath Amaratunga, the chairman of the University Grant Commission, for his commitments and leadership for the expansion of higher education sector of our country. Sir, we are highly honored that you are among us today to launch this conference. It is our privilege to welcome our Vice Chancellor, Professor Upul B. Disanayake. Sir, please accept our warmest welcome to this occasion. We would like to thank you for taking your valuable time for gracing this occasion. Our eminent keynote speaker, Professor Parakrama Karunaratne, I am greatly honored and pleased to welcome you, sir, on behalf of PGIS. Your acceptance of our invitation to be the keynote speaker is highly acknowledged. I would like to express our warm welcome to Professor Saluka Kodituakku, Dean, Faculty of Science, and Deans of other faculties, Director of Postgraduate Institute of Agriculture, Professor Dematavava, Director of NIFS, Professor Saman Senavira, for your active participation in this occasion. I would like to present my compliments to all academic colleagues and administrative officers of this university and PGIS who participating in this opening ceremony. Actually, without their support, this event would not be a reality. With tremendous pleasure and utmost gladness, I extend a warm welcome and appreciation to all invitees, researchers, delegates, and other stakeholders to the RESCON 2020. I am pretty sure that many of them are participating in this event through online platforms. I would like to close my speech by expressing my sincere wishes for the success of Congress and for all participants to discover new opportunities in the still growing area of scientific research. Thank you very much indeed.
Thank you, sir. The Faculty of Science of the University of Peradeniya has been an inseparable partner of the PGIS from its inception. I heartily invite Professor Saluka Kodituakku, the Dean of the Faculty of Science, to address the gathering.
Good evening, everybody. Professor Sampath Amaratunga, Chairman, University Grants Commission, and the Chief Guest of the day, and Professor Pulbi Disanayaka, the Vice Chancellor of the University of Peradeniya, and the guest of honor, Professor Parakrama Karunaratna, Deputy Vice Chancellor, the University of Peradeniya, and the keynote speaker of the Congress, my fellow deans, directors of the institutes, chairmen and the secretaries of the boards of studies, the registrar of the University of Peradeniya, bursar of the University of Peradeniya, academic staff members, administrative staff members, non-academic staff members, invitees, ladies and gentlemen. Though the Director PGIS welcome you all for the RESCON 2020, as the Dean of the Faculty of Science, I would also like to welcome you all for the 7th PGIS Research Congress, RESCON 2020. Though I am the Dean today, I still remember in 2014, I was the first RESCON chairperson. As the first chairperson, I am glad to see the progress of this annual research congress. Director PGIS talk about the academic staff strength of the PGIS, but I have to say that PGIS is a national institute, but it is the research arm of the Faculty of Science. As a result, majority of the academic staff members, including teaching staff and the supervisors of the research projects, mainly drawn from the Faculty of Science. And in addition to that, since 1996, academic staff members of the Faculty of Science, they work very hard in order to bring the PGIS or Postgraduate Institute of Science to the where it is today. The many directors work hard, their dedication and then the commitment also result in the success of the development of the PGIS. Even today, academic staff members of the Faculty of Science, they take all the hard works in order to organize almost all the activities of the PGIS under the leadership of Professor Pitawala. And not only that, academic staff members of the Faculty of Science, they carry out multidisciplinary research in collaboration with the other faculties of the University of Peradeniya. As we all know, University of Peradeniya is the only university having all 9 faculties. As a result, our researchers carry out multidisciplinary research that leads to the innovative outcomes. And mainly I can remember many of our department staff members, that is the computer science and statistics, they also carry out the collaborative research with Faculty of Engineering, Faculty of Dental Science, Medicine, and so forth. And now even it has been expanded to carry out multidisciplinary research with the Faculty of Art and the Faculty of Management as well. And then I also express my gratitude to the PGIS for bringing all the academic staff members of the University of Peradeniya under one umbrella in order to carry out the nationally important research projects. Even during this COVID pandemic period, our researchers initiated many research projects. The other partner of the PGIS and the Faculty of Science is the National Institute of Fundamental Studies, NIFS. And then the NIFS also, many of the young researchers, they registered at the PGIS and they carried out their research projects under the supervision of NIFS staff as well as the academic staff of the Faculty of Science. At the same time, I must express my gratitude to the staff members of the Faculty of Science. While carrying out their teaching responsibilities associated with the Faculty of Science, they work hard in carrying out the research activities, collaboration with the PGIS. And then also at the same time, I must thank the Director, Postgraduate Institute of Science for helping us in various ways to develop our laboratories and then also to provide some teaching aids. Then finally, once again, thanking everybody for actively participating in this wonderful Congress, though I did not expect this number of participants under these circumstances, but anyway, then thank you very much for actively participating. And also, I hope next 2 days, though this time Congress is different than our regular conference, because in the regular conference are not conducted using ICT facilities or video conferencing facilities, but this time we had to change it. But even it is the case, I hope the activities of the Congress will be a grand success, and then also expecting that the PGIS Congress 2022 will be a platform for experienced as well as young researchers to showcase their research talents. And finally, I wish the Congress 2020 a grand success. Thank you very much.
Thank you, sir. The University of Peradeniya, the queen among all the universities of Asia, is the bedrock upon which other institutions grow and thrive. We are grateful that the Vice Chancellor of the University of Peradeniya, Professor Upul B. Disanayake, is here as our guest of honor. It is my pleasure to invite Professor Upul B. Disanayake, the Vice Chancellor of the University of Peradeniya, to share his thoughts with us.
Professor Sampath Amaratunga, Chairman of the UGC, Director PGIS, and Chairman of the Organizing Committee, Director of NIFS, Deputy Vice Chancellor, all other directors, Dean of the Faculty of Science and all other deans, participants, ladies and gentlemen. First of all, I would like to take this opportunity to thank and welcome our Chairman UGC for being with us to grace this very important occasion at this very difficult hour. I know the amount of work he had today in Colombo, facing 6 court cases. Somehow he came to participate, and that is an encouragement for all of us. Thank you very much, sir. And at the same time, all of the dean had an opportunity to meet and discuss our issues pertaining to the university development, and we are really happy for your favorable answers. We hope that the outcome will be very successful. At the same time, I want to thank and appreciate the activity of the director and the chairman. They were very excited during last couple of weeks thinking that COVID will prevent them. But now you have proven that COVID can't prevent you all. You have organized this in a very successful way as usual. And then my appreciation and it should go to the presenters who are going to make about around 300 presentations tomorrow and within these 3 days in this special different type of different mode of presentations which we have not used in the past, and that this is how we must use our technology to move forward. We don't know when this is going to be end. Therefore, whether we like or not, gradually we have to adapt with the situation and work accordingly. This is not just to stop anything, but we have to move forward. That, of course, we must understand. Again, if you are going to, tomorrow, this is going to be a good platform for those scientists, especially young scientists, to discuss their new innovations, findings, and new research ideas, to exchange their views, ideas with a group of intellectuals in this platform. At the same time, I believe that new innovations and findings that you are going to present tomorrow, maybe, some of them may be helpful for us to address issues, burning issues of this society or in the country or in the world. Because this complex world is facing problems day by day, escalating problems day by day. But you are the people who can find out answers for these escalating, burning issues of these societies as scientists. I think Peradeniya scientists, no doubt, they can take that challenge and find out answers for most of these burning issues of the society which we face day by day, maybe the weather, temperature, climate, and so on and so forth. And I am not going to extend my speech more than that. Before wrapping up, as I mentioned, Chairman Sir, as I already have highlighted to you, within the last 3-4 years, I know there is a very good relationship with these postgraduate institutes and the university. Even though they are university institutes, the relationship between the faculties and these institutes, including PGIA, PGIS, PGIS, we were not happy about the relationship that he had in the past. But under the new, under these present leaders of these institutes, they are maintaining a very, very cordial relationship, collaboration with their own faculties. And I really admire that. I must appreciate both, all directors at present. But I'm not sure in the future, when these gentlemen are not there and I'm not there, what will happen for this association. Therefore, this moment I would like to request you. Now there is a new change coming in for our act. In that, you have to think of this and specifically accommodate this in such a way that we can maintain a very healthy relationship with faculties and between faculties and these PG institutes, because the fourth one is going to come very soon, Postgraduate Institute for Medical Sciences to Peradeniya. We have already advertised the director post. Therefore, we need a very concrete framework to work together for mutual benefits, not one direction benefits, but for the mutual benefits. Having given that thought, and I know that our Chairman Sir has got involved with the new designing of the new act, and therefore I request you to please pay attention to this one. Having said that, finally, I would like to take this opportunity to wish you all a very successful enjoyable sessions in coming 3 days. Thank you very much.
Thank you, sir, for your insights and your encouraging words. We are always thankful for the guidance and support you continually extend to the PGIS. Now, before starting the next portion, I would like to invite all the dignitaries at the head table to sit in the auditorium for the next presentation. And as we always go, we go higher. The University Grants Commission is the apex body of the university system of Sri Lanka. The institution works tirelessly to make Sri Lanka the knowledge hub in the region and a leader in higher education. We are honored to have the person who's leading the UGC as our chief guest tonight. It's my honor to invite Professor Sampath Amaratunga, the chairman of the University Grants Commission, to address the gathering.
Good afternoon. Vice Chancellor, University of Peradeniya, Professor Upul Disanayaka, Director, Postgraduate Institute of Science, Conference Chair, Professor Rohan Fernando, and all Deans of Faculties, Mr. Aponsu from Ministry of Higher Education, and all colleagues from academia. First of all, let me thank Postgraduate Institute of Science inviting me for this prestige occasion. Coming down to Peradeniya for an international conference like this caliber is very much fruitful. I was invited by my very good friend Professor Rohan several times, and also the the director of the Postgraduate Institute of Science. I have requested, please don't come. Don't send me any invitation, just a WhatsApp message is enough, because I am part of academia. And our clear intention is the development in our field. So all know that I was the president of FUTA several times, several years ago. So I don't feel that I am a regulator. I don't feel. If you are coming from such background and if you feel that you are a regulator, it's wrong. It's wrong. So before coming here, I was studying what are the circulars that we have issued so far by the University Grants Commission. There are 2,000 or close to 2,000 circulars, but I have found only 2 circulars issued in the area of research in this country. One was actually drafted by Professor Rohan. That is to issue the circular for research elements. At that time, the FUTA made this and he wrote the draft, and we had so many things, and that was signed by the chairman at that time. The final one was developed by Professor Niranja from Peradeniya and signed by a chairman. These are the two circulars actually that UGC have circulated so far under research. There are so many circulars regarding research payments, but my concern is not that. My concern is the research development. Even this conference, if not few people, we don't have this. We cannot have a conference like this. Everywhere in our system, we have conferences, we have publications, that is for the interest of few individuals. They earn money, they collect money, they spend money, and they have conferences. Even our journals, the situation is same. That is why all universities have now stuck at one place. When it is come to ranking, now we cannot push universities from this place onwards. We are now totally blocked. Totally blocked. So as an economist, I am actually studying with all these research data. I continuously request from my colleagues, just tell us how Peradeniya is doing. I will show you some data today. I have brought some data comparing 6 universities, how we work actually. Okay. Look at this. This is all Scopus information. Sri Jayawardhanapura, Colombo, there is a mistake. Sri Colombo, University of Kalaniya, Moratuwa, Peradeniya, Jaffna and Ruhuna. First 6 universities. Scopus. You can see Peradeniya and Colombo, they are doing well. They are reaching 500 Scopus papers per annum. Professor Rohan categorically told me that we are not going to have any garbage in this conference. He said, we are going to assure the quality of papers. That is why the director, Postgraduate Institute of Science, clearly mentioned, these people are with us. Then we can just imagine the quality that you are going to maintain today. So look at Moratuwa, they have reached 600 because they conduct IEEE conferences. After the conference, they send 100 papers, 150 papers to IEEE, which will automatically grab to this corpus. and they select majority from them. Otherwise, they cannot achieve that height. Just for your information, I have given this to another place also. You can see last 5 years, the rate of change 68.5 every year, 43.1, 33.7, and Peradeniya 18.8, because you are at the climax. Other places, we have enough space for development. Once you achieve the climax, you are stagnant. Then you need a huge support or a structural change for the next step. Otherwise, you cannot go into that stage. Everybody will come to this level and stagnant here. So these are few statistics, very interesting. Look at this. This is also from Scopus. Who is doing well? Which faculty? Which area? Where those people are living? Do they have computers? Do they have laboratories? Do they have publication fees? Anything? Very interesting. Without this insight, I don't think we can issue circulars. Without this one, we cannot develop research for tomorrow. You can see Kelaniya, Colombo and Jayawardhanapura. University to university, their speciality is different. Maybe you have same faculties, but university to university, totally different. And we don't have a mechanism. Maybe after 65, we have to say bye-bye. Even your H index is 25. We don't have any mechanism. We don't have any mechanism to retain researchers, at least on contract basis, not in the permanent cadre, not to give sabbatical leave, not to allow free trips, but at least to work in our laboratories with our postgraduate students. We don't have that, at least. So we are now working on this matter. My opinion is there is no one to get the responsible of research in this system. Our education standards are very high. Very high. Maybe we are above others when it is how to have a model for research. I have opened up this at the commission level several times, asking everybody to give me your opinion, at least to set up a faculty for research. Faculty for research, not only for medical science and engineering, or management, or combinations. So at least 10% or 15% from the current or the capital grant we can easily use for research. When I was at Sri Jayawardhanapura as VC, we have developed more than 500 million fund. Every year, 150, 200 million we used to invest for research. In our annual increment form, most of the universities does not have a place to mention the research that you have conducted within the year. Even if you have research or not, you will definitely get that 750 rupees at the end of the day. to get 750 rupees or 1000 rupees, you don't need to produce research. So many things that actually we have to debate. So that is why I actually at the outset thank Peradeniya inviting me. Thank you so much again. Otherwise, I am alone. I am with 4 members at the commission and full of administrative staff. As a model, I see there are some weaknesses. For a discussion like this, there is no Senate, there is no supreme academic place to discuss. So I have to go out and meet people in this caliber and tell my problems and bring back feedbacks, and then only I can send circulars. I am not fond of sending circulars. So this is the situation, and these are the number of circulars. Payment for research, various activities. Final level, I will show you this. It's there. Can you take this? It's in the... Yes. Shall I go another? Shall we check another one? Okay. Next. Yes, not the next slide. The next file. Okay. This is the latest. This one is the latest, QS. I don't know whether you all have received this, Peradeniya number 1. So Peradeniya is the most eligible place for this research development because this is the only residential university. All scientists can stay here and work 24 hours. Only this university have this facility, not in other places. I will show you another diagram and finish my work. Web of ranking. Yeah, second one. It's all right. No problem, right. All right. You have data from 2014. You can see all big universities are now stuck. You cannot just move from this level. See how many years that you have to experience to move 250. But in the latter part, they move by 1,000, 2,000 and 3,000 every year, other small universities. But we are stagnant. Since only few individuals, those who have this interest, run the whole research programs in our system. So give me your opinion, and we are in a position to change the mindset and to set up maybe faculties, maybe institutes responsible for research in each and every place. So I don't want to take much time. Thank you so much, Director, Postgraduate Institute of Science, Vice Chancellor and Conference Chair inviting me for this prestige occasion. Thanks.
Thank you, sir. Please remain at this podium, sir. We have the next event. Thank you, sir, for your kind words and your service to the higher education. I'm sure this event would spark the conversation of how we should look into the future of research and development. To honor your presence, the PGIS would like to present a token of appreciation. I invite Professor HMTGA Pitawala, the Director of the Postgraduate Institute of Science, to come up to the stage to present the token of appreciation. Thank you, sir. And we have a special event planned for today. For the first time in the conference history, we are launching the RESCON proceedings and the PGIS research highlights online at the inauguration ceremony. For it to be accessed to a wider audience and to disseminate the knowledge that was generated in this research congress. To officially launch the proceedings as well as the research highlights, I again warmly invite the chairman of the UGC, Professor Sampath Amaratunga, again to the podium.
ఎందుకు అప్పుడు డైరెక్ట్ అయ్యాను మేకని మేక
You can download proceedings.
Okay.
You should again go to these links and you can download files.
Thank you, sir. Now, we come to the pinnacle of our event today. I invite Professor Rupika Rajakaruna, Professor in Zoology, to the podium to introduce our esteemed speaker, keynote speaker of the day.
The dignitaries of the front table, now they're in the front table, the distinguished guests, ladies and gentlemen, good evening. It's a pleasure and it's my privilege to introduce the keynote speaker today, Professor Parakram Karunaratne. Professor Karunaratne is not a stranger to this community. He's a senior professor in zoology and is currently the Deputy Vice Chancellor of the University of Peradeniya. Professor Karunaratne received his secondary education from the Royal College, Colombo. He entered the University of Peradeniya and obtained his bachelor's degree in zoology honors in 1984, and later his master's degree. Then he obtained his PhD from the London School of Hygiene and Tropical Medicine at the University of London in 1994. He held many academic and administrative posts at the University of Peradeniya as well as elsewhere. He holds the chair of zoology since 2001 for almost 2 decades. He served as the dean of the faculty for 2 terms. He was the director and senior research professor at the National Institute of Fundamental Studies, the NIFS, at Hantana, and then he also served as the head of the department zoology and was the proctor of the University of Peradeniya. He has held many numerous visiting positions and fellowships. He was a Wellcome Trust Research Fellow, a visiting research professor at the Liverpool School of Tropical Medicine in the United Kingdom, the visiting research fellow at the School of Biosciences, Cardiff University in Wales, a member of the DDT expert group of the WHO UNEP Geneva, Switzerland. He's an elected fellow of the National Academy of Sciences since 2006. He's also an elected fellow of the Royal Entomological Society since 1997. So Professor Parakrama's main area of research is medical entomology. He has written and co-authored over 90 peer-reviewed full papers having over 2,700 citations and with an H index of 27. His pioneering research on mosquitoes and other vectors of diseases with special emphasis on insect control methods and mechanisms of insecticide resistance have received local and global attention and many awards. And to name few, the General Research Committee GRC Award by the Sri Lanka Association for the Advancement of Science, SLAS, Sri Lanka, and the CVCD Excellence Award for the Most Outstanding Senior Researcher in 2016 by the UGC, Sri Lanka, Westergaard Fredson Award for Outstanding Research Contribution by the Indian Council of Medical Research, Bernard Soisa Memorial Award, he received a gold medal for that, for Outstanding Scientific Research, this is by SLAS. Hiran Tilakaratne Award for Outstanding Postgraduate Research awarded by the UGC Sri Lanka. And in 1999, he received the Young Scientist Award by the World Academy of Sciences, which is known as the TUAS, Italy. And to date, he has 17 presidential awards and several NRC Merit Awards for his research and publications. He has also received the NSF, the National Science Foundation Merit Award for the best scientific research in biology in 1999, and the Wilson Peris Memorial Award in 1989 by the Sri Lanka Medical Council. So for all these reasons that we have Professor Parakrama here with us today delivering the keynote speech. He will be talking on his favorite subjects, the mosquitoes. Ladies and gentlemen, please welcome Professor Parakrama Karunaratne.
Thank you very much, Professor Rujpika Rajakaruna, for your kind words. UGC Chairman, Sir, Vice Chancellor, Sir, dear colleagues, Registrar, Bursa, and other non-academic staff members, Dear students, ladies and gentlemen, so I'm sure this is a very familiar topic to you all, the mosquitoes. Well, we all know that it's a biting nuisance, and at the same time, it is recognized as the biggest killer on the planet. That is because they transmit very, very dangerous human diseases in humans. Due to mosquito-borne diseases, more than 1 million deaths occur worldwide annually. More than 1 million deaths means it kills, these diseases kill one person in every 30 seconds. In every 30 seconds, one person on this planet is killed by these mosquito-borne diseases. So that is about 3,000 per day. And we have, you are familiar, I'm sure, with 4 diseases we have in Sri Lanka. We had, because this malaria is no longer existing. Filariasis also, they say it is no longer there. So the WHO has given certificates for those 2. But we don't know when it is going to be reintroduced, because the vector mosquito is with us. And it is there in the neighboring India. So like India, the COVID came, these things can also come. If they come, the parasites come, the mosquitoes are willing to transmit all the time. And the Japanese encephalitis, we have about 10 cases per year now. It has been reduced. Once it was very highly, it was 2 epidemics in Anuradhapura in late 1980s. And dengue is the most dangerous one to date. I think close to 30,000 cases in Sri Lanka, and about 32 deaths in Sri Lanka this year. But when you take the world, there are a lot more other diseases transmitted by mosquitoes. Now malaria is the, when you take the world, this picture, malaria is the highest killer. And you can see in Africa, the cases for last 10 years, more than 200 million, around 200 million for last 10 years. In fact, this has been happening over the years for a long time. That is why we say biggest killer. COVID is there, we know. It has become the biggest killer nowadays. But a vaccine will come in another few months, I guess, and then it will be gone. But the mosquito is there, and if the dengue is gone, the new one will come. Because when we were children, actually, dengue was not there. It was, the first case was detected in Sri Lanka in 1962. Likewise, new things can come because mosquitoes are there to transmit. Now in Sri Lanka situation, malaria was once the most very dangerous disease in Sri Lanka. In the year, this is a 100-year profile here from 1911 to 2011. In the year 1935, there were 5.5 million cases in Sri Lanka, and the deaths were 80,000. In 1934-35, that per year, deaths we got 80,000 deaths. And then after the Second World War, we had DDT, so that it went down, and then so it went down, and then with the DDT we could control. Then the mosquitoes came with resistance to DDT. It again went up. Then we replaced DDT with malathion. It went down, and then again malathion resistance came, and then we introduced pyrethroids. It came down, and then finally by 2015 we could get rid of malaria, and now 2016 WHO gave the certificate that Sri Lanka is a malaria-free country. Now the thing has gone from malaria to dengue, because now when the developments comes also, now malaria mosquitoes, they breed in the riverbeds and also marshlands. Now all those things are now being, forest areas are now converted to urbanizing the rural areas. So these create a lot of very favorable breeding sites for Aedes mosquitoes. That is the vector of dengue. So that is one reason why we are getting high dengue, because there are a lot of breeding sites around with new constructions. And as I told you, the number of dengue cases is very high. In 2007 is the highest dengue cases we got, 186,000 dengue cases with 440 deaths. So that is a very high number. And then the other danger is that mosquitoes can transmit diseases from animals to humans. In fact, the dengue also, it is believed that about 800 years back, the dengue virus transmitted from monkeys to human in Africa. So all the bad things we put to Africa, that is another thing anyway. That is how the scientists believe it has come to humans, from monkey to humans in Africa, then subsequently spread throughout the world. So the animals to humans, this is why I said that even we control dengue, we don't know what is there to come in tomorrow, right? Now, this is a frog. Now, if you can see the backside of this frog, there are more than 20 mosquitoes. Now this one, there are more than 20 mosquitoes are biting the backside of this poor frog, right? If you count. So this photograph actually was taken by Professor Priyanka De Silva from the zoology botany, this pond, backyard pond, at about 7 p.m. She has taken this nice photograph. And then also, you can see in birds also, our Dilaj Chaturanga from Chaturanga, I borrowed this. You can see the mosquitoes are biting. And also we did a good study in the forest areas in Sri Lanka, just in different heights. Vertical distribution of mosquitoes we assessed, forest ground and understory level and canopy level. Canopy level actually we wanted to see whether, because roosting birds are there, bird nests are there, so there are specialized bird feeders or ornithophilic species you find at the canopy level, and some are in the ground level. At the same time, there are mosquitoes like Culex quinquefasciatus. They are available in all the levels. So they are the most dangerous one, and also we found a lot of parasites in these mosquitoes. So they are capable of giving us these diseases at any time. So that these are some of the recent papers we published on ornithophilic mosquitoes and frog-biting mosquitoes. Now I will give you some sort of introduction about the mosquito life cycle. I think you are very familiar with this. Female mosquitoes lay eggs. Now this is especially Aedes life cycle is there, so that Aedes mosquitoes breed just above the water surface on the substratum. So you can see dark dots here. So these are the eggs. So even now, if you want to clean the water containers, if you throw away the water, that is not enough. You have to wipe out the inner surface. You have to just rub the inner surface of that, otherwise the eggs are going to be there. Now the bad thing with these eggs can survive during the dry periods. In Culex and Anopheles, the eggs also die when during the dry period. But here, they can wait until the next rain comes. So that is why we have to make sure eggs are not there. And then they can hatch, and 2 to 3 days, if the conditions are favorable, larvae comes. There are 4 larval stages, and then it takes about 45 or 1 week, and then pupa comes, and this pupa, after 1 or 2 days, then the mosquito comes. Okay. Now this is just to show the size of the eggs of Aedes mosquitoes. So this is a pencil tip is there just to show the size. relative size, and this is the shape of that egg under the dissecting microscope. And this is, Aedes lay eggs singly, but Anophelines, and this is a Culex mosquito, a Culex lay eggs like egg rafts. So these are whitish in color, mean when they lay the egg, it is very soft. That otherwise they can lay eggs, right? So it's very soft, and so that it can come through the vagina, and they lay the eggs, and with the atmospheric, when it is exposed to the atmospheric air, they get brown or dark, black in color. They lay eggs on water surface. Then from that, larvae comes. These larvae breed atmospheric air. So they have a siphon here, and through this they get the atmospheric oxygen. So you can see when the water is settled, they are there. If you disturb the water, they go down and come back to water surface. That is why if you apply oil layer, they can't breathe and they die, right? The very old days, before these insecticides comes, people used to use oil and powder-like things to prevent, to kill larvae. And then these Mansonia larvae, very smart. This is a root of a water plant, aquatic plant. They just put their siphon into the air spaces of the aquatic plant and get oxygen. So these are Mansonia larvae attached to roots of water plants. Now this is the pupa. Pupa, 2 to 3 days, the mosquito comes out. Right. This is the very first thing they do when they emerge, mating. Now when you look at this couple, this is the male. Male is usually smaller than the female. Male is easily distinguished by these two antennae. They are bush-like, lot of hairs on the antennae, not like in female. Lot of hairs are there because they are very reactive to pheromones given by female. They come closer and synchronize their wing beating, and they mate. Now when they mate, male transfers sperms to the female through the tip of the abdomen. And when the sperms come to the female store it in a sac called spermatheca, so that female doesn't want a male any longer. After that, that is it. They don't have to meet a male again and again to get sperms. She can store sperms in a separate sac, and she lays eggs several times. When they lay eggs, when the eggs are matured, she puts some sperms from that sac on top of those eggs and fertilize. So that she can store the sperms and then can fertilize. I hope luckily human females can't do it, right? And so this is then, the female, once they get the sperms, once the eggs are matured, then what they have to produce the egg sheath. To produce the egg sheath, they need a lot of protein. To get that protein, they need a heavy protein diet. For that is why they are looking for this blood meal. They are coming to you to take the blood meal to get this protein diet. Now they have these mouth parts. These are mouth parts, especially they have adapted to make a roll down to make a pipe-like thing. This is the proboscis through which they suck the blood. And this is again a part of, there's a stylets of maxillae, there's a pair of mouth parts. These are specialized to pierce the skin, and then they suck the blood, right? Now these stylets, this is the one you use to pierce the blood. If you look at the scanning electroscope, microscope view of that, you can see teeth on that. They are well adapted to go into the skin, right? Pierce the skin. Then they suck blood. Okay, when they suck blood, they put first two things. One is anticoagulant they put, that is to stop clotting, because when they suck, if the blood start clotting in the proboscis, they will have problems. So anticoagulant compounds they send, and also anesthetic compounds they say. They give a local anesthesia. So this is a kind of operation they are doing. They are just not taking blood. So that local anesthesia is there, you don't feel that they are biting, right? And so this is the salivary gland. They put saliva before they suck blood. In this saliva, anticoagulant agents and anesthetic compounds are there, and also with this blood, the parasites or viruses in this mosquito go and accumulate in the salivary gland. So that when they put saliva, they just go to that bloodstream. That is how the blood is getting this, all the pathogens. So this is the mechanism. And then there are different terms: anthrophilic, zoophilic, some are coming to humans, some are going to animals. Endophilic, exophilic, some like to bite indoors, some like to bite outdoor. They bite you and go out. Now another thing is this spraying. When we were children, or when malaria was here, people, the health workers came and sprayed your walls with DDT or malathion. Now for dengue, they don't spray, because the Anopheles mosquitoes, when they take blood, they stay inside the house. They rest on the wall. So that is why it is effective against malaria. But it is not effective against dengue, because dengue mosquitoes don't stay, right? They either they run away, or they have hiding other places under the desk or the clothes, right? So that wall spraying is not effective. Diurnal, nocturnal, some are daytime biters, some are nighttime biters. That is why the bed nets are not effective for dengue, because they bite you daytime. But it is very effective for Anopheles malaria vector, because they are nighttime biters, feeders. Their flight range is about 2 kilometers, roughly 500 meters, up to 2 kilometers. If there are strong wings, that the wing can carry them for further. And the lifespan is about 1 to 2 weeks. The female mosquitoes can live little longer than this. They feed on sugary liquids like fruit juice, plant juices, and the blood meal for egg development. I told you the blood, for the egg development, they need the blood meal. So females lay egg 2 to 3 days after the blood meal. Several gonotrophic cycles, that means the females can lay several egg batches, 3, 4, 5 batches of eggs per lifetime. Then the mosquito diversity. There are about 3,500 mosquito species in the world, and about 160 species in Sri Lanka. So there are 160 different species of mosquitoes in Sri Lanka. And out of this, actually about 20 species can transmit diseases. Can mean, so far we have identified, established vectors are less than 20. So we had done extensive work on this DNA barcoding of mosquito species. This is very important because this is how the mosquitoes are identified. This is the thorax, back and this side thorax of a mosquito. You can see these scales, black scale and white scale. This is again, this is the backside thorax of Aedes aegypti mosquito, and likewise this is Anopheles. In the wings you can see these are all scales. So what happens, the thing is, when you collect mosquitoes, they come close to each other. When you put them together, then these scales are gone. It is very difficult to identify. So that this barcoding is really help, DNA barcoding, to identify, so that you identify different barcodes for different species. And also there are sibling species. Morphologically, you can differentiate. And it is very important for mosquito population genetics. I'm not going to go into detail into these technical things. And we have done about 39 mosquito species we have barcoded so far. This is a continuous project actually. And we have come up with very good publications recently. And this chemical control. I said that chemical control is the most effective control we have. There are several other control methods. I'm not going to talk on all those things. And this is wall spraying, this is the mosquito coil, this is bed nets. So bed nets in Africa, this is a very powerful method for against malaria. These bed nets are impregnated with insecticides, pyrethroids. So what they say, that dosage, even if a small one put it to the mouth and chew it, it is not going to be harmful. According to the WHO assessment, it's a very, very low dosage, but it will repel mosquitoes. Otherwise, when the holes are there, mosquitoes try to come inside. So when the insecticide impregnated nets are there, that repel the mosquitoes from the net. Even the holes are there, they are not trying to come inside. And then, yes, then fogging for dengue vectors and spray. This is for larval control. They spray malaria, they spray insecticides. Now when it comes to insecticides, we use organophosphates, heavily used like malathion, pyrethroids. Pyrethroids is it was introduced to Sri Lanka in 1994 and it is a very successful. It has a very low mammalian toxicity. And then carbamates. Carbamates we are not using much. It is separate, heavily used in the agricultural sector to control agricultural pests. And organochlorines. Organochlorines like DDT, we stopped using DDT in 1975-77 period because it is very harmful and pollute the environment. So that we stopped DDT using that. In fact, I am in the World WHO UNEP DDT expert committee. That committee, what it does it, even though this is very environmentally unfriendly, some countries still use DDT because that is the most reliable method they can use in those countries. 5 countries are there still actually: India, South Africa, Zimbabwe, Zambia and Namibia. India is the producer. They produce about 20 metric tons still. So what our job is there in that committee is to find alternatives for them and advise them to how to reduce these things. Reduce the usage of DDT. So we expect in another 5 years to get rid of this DDT completely. And then insecticide resistance. So this is the major problem with the insecticides. When you spray insecticides, they become resistant continuously when used. What happens inside the insect body? This is when you spray insecticides, this is the insect body. When it goes to the inside the body, and this is the target site is the insect nervous system. So before it goes to the insect nervous system, they have 2 methods. One method is insect enzymes can break down this insecticide so that no insecticides are not going to the target site. The other method is they can somehow escape from enzymes and go to the target site, but the target site is altered. It is not sensitive, mutated target sites. So the resistant mosquitoes, they have either large quantities of these enzymes or mutated target sites. We have studied these things extensively. We were the pioneers to study this work. Actually, we did this in collaboration with Liverpool School of Tropical Medicine mainly. And we are the Culex quinquefasciata from Sri Lanka. We got Sri Lankan mosquitoes, and we found huge quantities in these mosquitoes. The basic mechanism was gene amplification, so that in a susceptible mosquito you get 1 gene. In resistant mosquitoes you get 250 copies of that gene. So they produce huge quantities of these enzymes. So when they come into these organophosphates, when you spray, they come to the body, they can metabolize those things. These esterases, because organophosphates are esters of phosphoric acid. Now, we purified and characterized, and sequestration role mean you need large quantities because these esterases, within milliseconds, first part of the reaction they do, and for the second part of the reaction, to release the free enzyme, you need about 24 hours. so that whenever they see these enzymes, whenever they see the insecticide, just absorb it, and after 12 hours it is broken down. So to do that, they are working as sequestrants, so absorbents. They need large quantities. That is why they need large quantities. Then we raised antibodies for these enzymes, and using these antibodies, what we did was we sliced the mosquitoes and put this antibody, and then we could see where these enzymes are using electron microscopy, and we could locate, we could see that these enzymes they are under the cuticle, around the gut, and in the salivary glands. Actually, these are the places through which insecticides enter. So when the insecticide enter at the gate, actually there are a lot of enzymes. So that they can't go into the body, they break down the insecticide. So these things, we did a lot of work, a lot of publications came, and 2 landmark publications came, one in Nature in 2000 and one in Science in 2010. The other thing is, I said, they go to the target site, but the target site is altered because of mutations. We have done a lot of studies on these pyrethroid insecticides. The target site is the insect sodium ion channel, regulatory proteins. Those who have done biology should know that nerve membrane, if you take, the impulses are going because sodium ions are coming in, and once the impulse is gone, sodium ions are actively pumped outside and close the gate. Now these sodium channels, the sodium business happens through the sodium channels. Pyrethroids go and attach to these gates, so they keep the gates open. So sodium ions are always inside. That means the nerve is always in the excited stage. So because of the excited state, it comes to a coma and die in the insect. That is the mechanism. But then they have gene mutations. These insecticides can no longer bind to the target site. They can no longer bind to the sodium channel gates. So this is the molecular structure of the sodium channel. 4 domains are there, and we found mutations in these domains. And we found in several insects, Sri Lankan insects. So this is Anopheles subpictus mutation, Culex quinquefaciatus. The Anopheles subpictus is the malaria, Culex quinquefaciatus filariasis, Aedes aegypti, Aedes vector, Cymex hemipterus, that is the bed bug. This is an interesting story. Now there are 1 to 6 mutations in this. These bed bugs we collected from Ramanathan Nolan, Jayatilaka, and Akbar. We got this journal, Parasites and Vectors. The impact factor is about 3.4 SCI journal. And we had monitored it for a long time. So this is why when you spray pyrethroids, they don't answer, because they have 6 mutations in these resistant bed bugs. Right? So when you spray, all the susceptibles are getting killed, resistants are survived. Next generation comes from that resistant ones. So that ultimately they all get resistant. So Vice Chancellor sir, we spent a lot of money, and after a few months again the complaints came, a lot of bugs are there. So yes, and then the other one is the cattle tick, and the last one is the sandfly, right? So several publications came from. Then I'll move on to this insecticide fogging. We did, because the way they were just everywhere spraying. Nowadays, then when corona is there, we get the request to sterilize our house. Come and just spray these sterilizers to these places, because the neighboring house there was a COVID patient, and even the university we get. Now this is also like that. When the dengue case is there, they go and spray the whole area. So that is environmentally not good. We just tested this effect on pollinators. Actually, out of 3,884 knockdown insects, we did very scientifically and several sites and several replications. Out of 3,800 mosquitoes, only 31 mosquitoes. Out of 31 mosquitoes, only 2 were Aedes. So you kill those two, but you kill all the other insects. A lot of other insects are getting killed. This is a little bit of, because this is open air we did. So mosquitoes are there in some hiding places also. They are not coming into count. But anyway, in the direct study you can show that when you put the mosquitoes in these cage bioassays and expose to this fogging, and this is a very important pollinator insect. It's a bee, stingless bee. And when you put this one, it is also getting 100% knockdown, and after half an hour, 7% recovery. Mosquitoes, 100% knockdown, 12% recovery. Actually, it is more harmful to these bees, so that we did that study. So this shows that this insecticides is not a good choice to be used for a long time. So are we going to continue with insecticide-based mosquito control? That is the question, because we all know that insecticides are not good to the environment and to the human health and everything. Even though they say pyrethroids have very low mammalian toxicity, they have proved now they can long-term exposure can give cardiovascular diseases. Now there are two promising alternatives for dengue mosquito control. One is Oxitec sterile male technique, and the other one is Wolbachia infected mosquitoes. This Oxitec is the pioneer for this work is Professor Luke Alfay from Oxford University. Oxitec is the commercial arm of Oxford University. Wolbachia pioneer work was done, and he is still the person, actually, Professor Scott O'Neill. In this Oxitec, what happens is they put a gene to the mosquito, and that gene produces a protein, and mosquito die. So that is very simple. So what happens is, if you release that mosquito, they will mate with the females, normal female, but from this one, if they mate, then they die. So this is very simple. Then that gene is in the progeny, then they will die, next generation. But the thing is, in the laboratory, you have to grow this so that that gene had the thing that in the presence of antibiotics, this gene is not working. In the presence of antibiotics, this gene is not working. That means inside the labs, you can grow by putting antibiotics to the larval balls. When the antibiotic is there, gene is inhibited. Otherwise, gene is active, they kill larvae. So they came to Sri Lanka, and I am actually in the National Dengue Control Unit Technical Advisory Committee. So we had several questions. The problem is we have to regularly release these. When you release this first batch, then it will reduce the population, then again and again. And it is very, very expensive. Second method is Wolbachia. So this is the last topic I want to talk. And this is the endosymbiotic bacteria in arthropods and nematodes. So this is endobiotic bacteria live in these insects. So this was actually discovered somewhere in 1920, but recently only this interest came. So they pass from generation to generation, so that you don't have to repeatedly spray. They are just maintaining in the population. Now they have very good characters recently identified. When they are there, they don't allow other pathogens to grow inside mosquito. So when the Wolbachia is there, they compete with the virus, dengue virus, and they don't allow other pathogens to grow. And then also, they shorten the life of mosquito. That is also good. And also, in certain process, they cause embryonic deaths. For example, in this cross you can see, if the black is the normal one, red one is the Wolbachia infected one. So when these crosses, you get 2/3 in the next generation, you get 2/3 Wolbachia infected, 1/3 non-infected. So what happens is actually proportion of infected mosquitoes expand naturally over generations. Over generations this happens. Right. Then here's the one. Now the thing is, in for dengue, the major vector is Aedes aegypti. In Aedes aegypti, this Wolbachia is not there. So what they did, he is the person behind Scott, Professor Scott from Australia. They introduced this Wolbachia to Aedes aegypti and made a very stable line with all these efficiencies. And at the same time, we tested our populations for Wolbachia. This paper was published in 2017, and you can see we collected mosquitoes from 7 districts, and you can see in Aedes aegypti, as usual, and in the whole world this is the case, Aedes aegypti, no Wolbachia. Aedes albopictus secondary vector, 50% Wolbachia. Now we also confirmed, and we also said to the National Dengue Unit, look here, this is not a proper, strange, because they were very afraid. People are afraid when you bring something and introduce newly to a population, to our mosquitoes, because we don't know whether ultimately it can go to bees and it can go to others. But this is a naturally occurring type, so that they are also happy with our study, and then they started this. This is the World Mosquito Program. That is the one in Australia. What they do is, it is a not-for-profit initiative, and they produce these Wolbachia mosquitoes. They are against Zika, chikungunya, dengue, and yellow fever. In 2017, July, we, the dengue, our ministry signed an agreement with them, and now actually in this year, we released this in a pilot study in 2 places. Nugegoda we released these mosquitoes, and in District 1, Colombo Municipal, that is Mattakulia. Mattakulia, Nugegoda, we released these mosquitoes in this year. How did we do it? We didn't want to bring mosquitoes from Australia and release here. So what they did was, they took eggs from our mosquitoes and went there and reared our mosquitoes, Sri Lankan colony, and then crossed to the Australian colony. Next generation, back cross to the Sri Lankan mosquitoes. Next generation, back cross to the Sri Lankan mosquitoes. So this is how it is happening, back crossing, because they wanted to make the final genetic makeup of these releasing mosquitoes Sri Lankan genetic makeup, not the Australian one. Because we always have these restrictions to expose or to release aliens in this country. So that is why. So that always Sri Lankan mosquito male cross to Australian female, next progeny cross again to the Sri Lankan male, next progeny female cross again to the Sri Lankan. Ultimately, after 7 generations, you can say, see that 98.5% Sri Lankan mosquito genetic background is there. Likewise, they did, and then they brought, and then they do a quality assurance. In Australia, they make sure that they inject Wolbachia, Aedes virus, and see that virus cannot survive in these mosquitoes, and they test for their fecundity. That means how many eggs they lay, are they normal, whether they grow properly, and all those things. And also they check for other pathogens. So all those things they do screening, and they are brought to Sri Lanka, and they are then released here. They release here in capsules. They produce capsules like tetracycline capsule. They put mosquito eggs and larval food into that and make a capsule. So when you put those capsules into water-filled yogurt cup, it will rupture and comes out larval food and eggs. So that eggs will give the larvae, and then mosquitoes are released. So that they have done it successfully in Mattakulia and Nugegoda. In late June, they released in Nugegoda. Early August 10th, they released in Mattakulia, but then the COVID came, and that is the problem. So actually here you can see, this is how you should do it. This is the normal mosquitoes. Wolbachia mosquitoes, you have to release consecutively in these weeks, 12 to 20 weeks. When you, until the 70% of the population, you can check this by doing PCR, 70% of the population has Wolbachia, then you can stop, because it will go automatically and will come to 100%. right? So what they did was, they did start us after 2-3 weeks. I checked with them, and because of COVID, they can't go to the houses and premises, so they had to stop. Now it is abandoned, but soon they will start on this, and this is our ultimate hope for dengue mosquitoes in future. And if this comes, we can get rid of dengue if because once it comes to 100%, it will maintain 100%. So that is because it will not, because I showed you in process, they are getting more and more, right? So that is actually what I have to say about these mosquitoes. And this is actually, I have to acknowledge my research team, Professor Priyanka De Silva, mainly working on ornithophilic mosquitoes and frog-biting mosquitoes, and Dr. Tilini Veeraratne is working on DNA barcoding and other stuff, insecticide resistance. 2 PhD students, Nalaka Nugapola and Dilan Chaturanga, he's here actually, and several other postgraduate students. Liverpool, we had Professor Janet Hemingway. She was the former director of the Liverpool School of Tropical Medicine, and funding mainly came from National Science Foundation, National Research Council, University of Peradeniya, and from UK, Wellcome Trust. We got huge money from Wellcome Trust for our enzyme work, and the Royal Society UK. And thank you very much.
Thank you, sir. Please remain at this podium for a moment. I think the audience would agree with me that was one of the most engaging and relatable keynote speeches that I've ever witnessed. Thank you for the scientific perspectives that you laid on a familiar topic, sir. As a show of gratitude to our keynote speaker, the PGIS would like to present a token of gratitude, and I would like to invite Professor G.W.A. Rohan Fernando, the chairman of the RESCON 2020 Organizing Committee, to present the token of appreciation. Thank you, sir. To mark the end of the inauguration ceremony, I warmly invite the chairman of the RESCON 2020 Organizing Committee, Professor G.W.A. Rohan Fernando, to deliver the vote of thanks.
Good evening to everybody. Chief guest, Professor Sampath Amaratunga, Chairman of the UGC, and guest of honor, Professor Upul Bijisanayak, Vice Chancellor of the University of Peradeniya, Professor Parakram Karunaratne, Deputy Vice Chancellor, and the keynote speaker of our today RESCON 2020, Professor HMTJ Pitawala, Director PGIS, Professor Saluga Kodituaku, the Dean of the Faculty of Science, all other deans of the other faculties, and director of the Postgraduate Institute affiliated to the University of Peradeniya, and members of the Board of Management of the PGIS, and Registrar, Bursa, and Chief Medical Officer of the University of Peradeniya, all academic staff, administrative staff, and non-academic staff of the University of Peradeniya and the PGIS, and presenters who witnessed the ceremony to the YouTube, and other distinguished guests. The Postgraduate Institute of Science, University of Peradeniya, has organized its 7th Annual Research Congress. On the behalf of the organizing committee, it is a great honor and privilege for me to make the vote of thanks as the chairman of the RESCON 2020 and also the past student of the University of Peradeniya. First of all, I would like to propose a hearty vote of thanks to our chief guest, Professor Sampath Amaratunga, Chairman, University of Grant Commission, for gracing today's Congress and delivering the inaugural address. Thank you for your very interesting study on research and thought-provoking address I am sure that all the participants have taken note of your suggestions and will be discussing within their department and the faculties the action to be initiated at their level. I wish to thank the guest of honor, Professor Upul Vidisanayaka, the Vice Chancellor of the University of Peradeniya, for attending as the key guest of honor. His continuous support and guidance help us to organize this event despite having COVID-19 pandemic situation. Professor Karunaratna, the Senior Professor of Zoology, as the keynote speaker, for accepting our invitation to deliver the timely important topic with very short notice. RESCON 2020 has brought together a rich diversity of researchers from almost all the state universities, non-state universities, research institutes, and the industries to share ideas and new perspectives in a wide range of topics. Behind the success of the Congress lie the collective and untiring effort of Professor HMTGA Pitawala, Director PGIS, who is the live wire behind the organizing the RESCON 2020. Big thanks goes to Professor Saluka Kodituaku, the Dean of the Faculty of Science, to attend the ceremony and given us all the necessary advices, support from the human resources and the infrastructure facility without any hesitation. Thank you very much, sir. And all of the BOM members, and especially Mr. Aponsu, who came all the way from Colombo, thank you very much for your attending here, and all other chairpersons of the Board of Study who have helped us many ways, especially in the editorial work. I wish to thank the deans of the other faculties, directors of the Postgraduate Institute, director of the National Institute of Fundamental Studies, Professor Senavira, who attended this occasion despite having a lot of engagement in their faculties and institutes. I am happy to see the face of the former director, Professor Laxman Disanayaka, to witness this inauguration ceremony. I am sure he is happy to see this event as a person who built this institute since 1996. It is very pleasing to note that 230 research papers will be published out of 283 abstract and extended abstract, the highest number ever received for a RESCON here. At 30 virtual technical sessions using 4 video conferencing rooms over the next couple of days. Untiring effort from Professor L.R.K. Pereira, Editor-in-Chief, and Professor Namal Priyantha, the editor, the Elite Editorial Board, and the contribution of 174 reviewers with thorough and timely reviewing of the papers were very valuable to improve the quality of the abstract. Mr. Dilan Chaturanga contributed tremendously since the receipt of the application through online conference management toolkit until the completion of the proceeding volume. While regretting that the COVID pandemic has prevented our planned Congress at PGIS, we were excited about the opportunity to present to us to hold the first ever virtual PGIS RESCON in the next 2 days. Holding the virtual Congress will be the great opportunity for PGIS to explore new frontiers and early innovation, as well as using the digital and social media, using the pandemic to reach wider audience all across the Sri Lankan than the previous plan. I wish to thank 30 academic coordinators who are behind the scene to coordinate the virtual conference between the technical support team and the presenters. Mr. Kapila Bandara, Mr. Asanka Amarasinghe, Mr. Saluka Seniratna contributed technical support to build up the virtual technical rooms. All the staff who will give the technical support to the virtual technical sessions is greatly acknowledged. The organizing committee appreciates the honorary service provided by the session chairs for all 30 sessions. A vote of thanks may not be completed without the making the acknowledgement of the following personnel from the Faculty of Science. I think Professor Saluka Koditaku will be a very happy person because he has a very blend of youth and very talented youth as academic in your faculty. I think especially to Dr. Aswini Amarasinghe for comparing this inauguration session and all the hard work of the Media and Publicity Committee. Dr. Pasanga Mantilaka for secretarial work and bringing up several sponsorship. Mr. Prabhat Gunatilaka and Dr. Sajit Abesundara for initiating the online conference management toolkit and paid setting of the proceedings and the website. And Professor Sanjeev Malaviarachchi for completing the book on the research highlight. And Dr. Hemalika Abesundara for handling all the responsibilities of the virtual technical sessions. Professor Nanda Balasurya for all food and refreshment management. And Dr. BMK Premsiri for registration work, and Dr. Rachita Mendis for logistic arrangement. From the PGI side, I must thank to Dr. Champika Bandara for all the arrangement for inauguration ceremony, and to Mrs. Situmini Vijayaratna and Mrs. Siromi Rajapura, the assistant registrars of the PGIS, for logistic and decorations of the every part of the PGIS. And it is great pleasure to work with all of you. I wish to thank all of the staff of the PGIS, Security Division of the University of Peradeniya, Health Center, Maintenance Division, and others who helped to make this event success. The Congress would not have materialized without the financial support from University Grant Commission, Bank of Ceylon, State Mortgage Bank, Analytical Instrument, Hemson Private Limited. Thanks go to Sanduni Offset Printers for printing both research highlight and the conference proceedings. Video Makers Private Limited has given their valuable contribution to handle the inauguration ceremony and YouTube transmission. Thank you very much. And at last, but not least, my deepest appreciation goes to the authors who submitted their outstanding research papers to the RESCON 2020. I wish that the Congress will be highly stimulating, enlightening, and enjoyable to all of you and all of the participants who will join us to the YouTube today. Thank you very much.
My name is Mark Ramsdale. I'm the Director of Education in the Department of Biosciences at the University of Exeter. For many years now, I've worked with microscopes as a microbiologist. I really enjoy helping students learn about the microcosm around us. Today, I really want to show you around our new digital classroom, made up of connected Zeiss microscopes. My students are in the first year of the medical sciences. In this microbiology class, we are studying protist diversity. Protists are a diverse group of eukaryotic microorganisms that are neither plants, animals, or fungi. The majority are microscopic and unicellular. They are highly organized with nuclei and specialized organelles. To study protists, we use Zeiss Primus Star connected microscopes using phase contrast and transmitted light illumination. To view subcellular structures, we sometimes use dark field illumination. Comparing images with each technique is like creating a mosaic image, which gives my students even more insights. The interactive classroom offers me and the university a chance of giving a good educational experience to our students. For example, we can project an image that the student is seeing onto a screen. That way, everyone can see this microscopic image. On the other hand, I can constantly see what the students are seeing on my tablet. This allows me to help them even more. Both teaching and learning are much more fun in this environment. And when learning is fun, our students get better results, and that's what our university wants.
Thank you. And with that, we conclude our glamorous inauguration ceremony. Tomorrow and the day after, we'll be having